Hello, my name is Alex Carr. I'm a postdoctoral fellow in the Gibbons lab here at the Institute for Systems Biology. And today I'd like to tell you a little bit about a project that I worked on for part of my PhD, which was recently published in the journal Cell Systems. So imagine your gut microbiome is like a rainforest. Thousands of species interacting with each other. In the rainforest you have plants and animals, fungi and insects, each with their own specialized roles, and many of them interdependent on each other for their survival. The same is true in the gut microbiome. And when we clear cut the forest to extract resources, this can have unintended consequences. It can open up space for weeds to invade, and those weeds can make it more challenging for the natural habitat to be restored. And the same is true in the gut microbiome as well. When we take antibiotics, this can wipe out many of your native gut flora and open you up for the possibility for infections by pathogens. One of the most infamous of these gut pathogens is the organism Clostridioides difficile, or C. diff for short. One interesting fact is that many more of us are carriers of C. diff than you might expect. Some estimates suggest that as many as 40% of us might be asymptomatic carriers of C. difficile. While having C. diff itself isn't necessarily a problem, it can actually kind of live quietly in the background for many of us. Issues arise when you experience disruptions to your gut microbiome, either through antibiotic use or maybe through experiencing diarrhea or anything that really wipes out the diversity in your microbiome. This can create opportunities for organisms for C. difficile to bloom and can lead to very severe sickness, and in some cases death. Now, the standard treatment for C. diff infections, or CDI as they're known, is, as maybe you would have guessed, more antibiotics. While this can be an effective way to remove C. diff, it's akin to trying to remove weeds in the forest by burning down the forest. It doesn't quite make sense, and it can actually open you up for a cycle of reinfection that can keep you in a state of illness longer than you would like. And what we've learned through recent decades of research into this particular disease is that one of the most effective ways to combat CDI is restoration of your native microbiome. And one of the ways this can occur is through the use of fecal transplants, or FMT, fecal microbiome transplants. These are great and have been shown to be fairly effective for this particular illness, but there's still a lot that we don't know about this process, and you are introducing a fairly complex mixture of things into your body, and it's hard to know exactly what impact that can have. And that's where our team came in. We asked the question, can we better understand this process, and can we develop a predictive understanding that might allow us to design personalized treatment for individuals affected by this issue? To do this, we leveraged computer modeling. You could think of the approach that we developed as akin to a weather forecast. But instead of simulating weather patterns, we're simulating the metabolism of the gut microbiome. We built what we call microbial community scale metabolic models, or MCMMs. I know that's a mouthful, but the idea is fairly simple. It's essentially a digital twin for your gut microbiome. You feed the model the composition of your gut microbiome and an estimation of the chemistry of your diet, and the model then can predict how the bacteria in your gut microbiome are interacting. This approach allows us to forecast for your gut. Essentially, we can predict whether or not you're susceptible for colonization by C. diff or not. And so in some guts, you can say the person is fairly resistant or possibly not. And so leveraging this approach and a large compendium of publicly available data, we explored whether or not we could accurately predict an individual's susceptibility. And what emerged from this analysis was some interesting patterns. We found that across thousands of individuals that we assessed, some guts were totally resistant to C. diff colonization. In those situations, the native bacteria were predicted to be able to outcompete C. diff for the resources that it needs to grow. In other cases, we found that some guts were somewhat susceptible. C. diff could colonize and grow a little bit, but not much. In those cases, you might expect that C. diff might hang out in the background, but not really pose much of a threat. And then in a third group, we found that C. diff was able to grow, predicted to grow with very high rates. And in those cases, there were not very many competitors, and these individuals are likely the ones who are most susceptible for developing a C. diff infection. So that's great. And we found that these patterns were fairly consistent across many cohorts comprised of thousands of individuals. But we wanted to know whether or not we could also use this approach to validate or predict the efficacy of personalized treatments for these individuals. To assess this, we leveraged a probiotic cocktail that's gone through phase 1 and phase 2 clinical trials and is comprised of a mixture of 8 different bacteria and has been shown to be fairly effective in mice for outcompeting C. diff, and is also well tolerated in human populations. Leveraging this cocktail and our modeling approach, we were able to accurately predict when this probiotic could outcompete C. diff. And interestingly, we could also predict situations where it was not likely to outcompete C. diff, suggesting that while this treatment is very effective, there are cases where people don't respond to this treatment. This is a big deal because this suggests that in the future, doctors might be able to leverage a patient's gut microbiome profile to predict whether or not they are susceptible to infection, and possibly could then design personalized interventions for their illness. So where does this leave us? Well, for C. diff infections and for many other diseases, the standard of care treats these conditions with a one-size-fits-all approach. But our research suggests that individuals' gut microbiomes are fairly unique, and thus treatment should be unique as well. By creating these personalized digital models, we are moving towards a future where we can predict an individual's susceptibility and design personalized interventions to prevent infection. And moving beyond C. diff, this approach can be leveraged to predict all kinds of other types of functional capabilities of the microbiome, whether it be related to diseases such as diabetes or the production of anti-inflammatory molecules like short-chain fatty acids. This approach will be a powerful tool moving forward. In short, we're starting to treat the gut microbiome not as a black box, but as a complex ecosystem, one that we can model, predict, and most importantly, protect.